Next, we will move to the second part of this chapter regarding the organization of the DNA in chromosome. In this part, I will describe how the DNA will be packed into a structure known by chromosome. To reach the structure of the chromosome, رح نقطع ب different steps, guys. So let's describe this intermediate form between the DNA linear simple structure and the chromosome. This structure is known by chromatin. من اسمها guys إذا بنقول chromatin أو chromosome, chromo بالأول at the beginning of this term means colored body. But if you look at the chromosome or chromatin هتشوفون colorless. طب why the scientists call these bodies colored bodies if they are colorless? Because they discovered that these bodies can be colored using dyes, so they can be stained فيني لونهم using dyes. So chromatin or chromosome they are stainable materials in the cell nucleus composed of DNA and protein. What kind of proteins are associated to the DNA? Two types. في عنا proteins بنسميهم histones وعنا proteins بنسميهم non-histones. And both are very, very important in determining the physical structure of the chromosome. بلاهن ما بقدر حول ال DNA لchromosome. So I will describe the characteristics of the histones and the non-histones protein. I will start by the histones. Histones, guys, are considered as the most abundant proteins in chromatin. أكثر بروتين بنشوفها بالدي ان اي هن الهيستون. They are positively charged. Why? Because they are composed of basic amino acids. وليش نحن بحاجة لبasic plus positive charge بالهيستونز؟ Because the DNA is negatively charged. بتعرفوا الدي ان اي مؤلفة من نيوكليوتايد وبالنيوكليوتايد في عندنا phosphate group اللي هو negatively charged. So if the DNA is negatively charged and the histones proteins are positively charged, plus or minus شو بيعملوا guys? Attraction. So هيك فينا نلقط البروتين على DNA. 5 types of histones are present, known by H1, H2A, H2B, H3, and H4. Okay, so 5 types are very important in chromatin packing. In addition, other than the histones proteins, you can find non-histones proteins. أكيد they are far less abundant than histones. أقل بكتير من الهيستون. They are negatively charged because they are composed of acidic amino acids. These non-histones plays roles also in the process of DNA replication, DNA repair, recombination, and transcription. So now we need to move, guys, to the chromosome packaging. How we can convert the DNA double helix, the linear simple one, into a packed structure, condensed structure known by chromatin and then the chromosome. So رح امشي أنا وياكم step by step in the level of DNA organization or chromosome packing or chromosome compaction. So I will start first, guys, by the histones protein. First step. تتذكروا قبل شوي شفنا 5 types of histones, guys. رح اخذ منهم only 4 types اللي هن H2A, H2B, H3 and H4. 2 of each of H2A, H2B, H3 and H4 will assemble together. So إذا أنا عندي 4 types times 2, so we'll have a total of 8 histones proteins. 8 histones proteins, guys, will be surrounded by 147 base pair segment of DNA wound around the 8 histones protein 1.65 times to form a structure known by nucleosome. So رح ارجع عيد على هيدي الفيجر guys. So باخد ال 8 histones اللي هن 2 each of H2A, H2B, H3 and H4. so بيعمل لي total of 8 histones. ورح لف عليهم 147 base pair of DNA. كم مرة بدي لف هال DNA؟ 1.65 times. Okay? To form a structure known by nucleosome. So what is the definition of nucleosome, guys? The nucleosome is defined by 147 base pair of DNA wanted around 8 histones 1.65 times. هيدي nucleosome وهيدي another nucleosome. The first nucleosome is linked to the second nucleosome by a linker DNA. And this structure, guys, is similar to beads on a string. كأنه خرز معلقين بخيط. So each nucleosome represents the bead, okay? Will linker DNA represent this string linking the first nucleosome to the second one. The dimension of one nucleosome, guys, is 10 nanometer. So that's why the structure of the beads on a string, nucleosomes linked together by linker DNA, is known by 10 nanometer chromatin fiber. So the 10 nanometer chromatin fibers, guys, represents the beads on a string and represent the least compact form seen inside the nucleus of the cell or of the eukaryotic cell. Next, هلا بدنا نقطع one level of packaging greater than the 10 nanometer chromatin fiber. إذا تتذكروا, we used 4 types of the histones proteins, H2A, H2B, H3, and H4. We still have the H1 histones protein ما استعملناها. So by the addition of the H1 histone protein, you will move to the next level of chromatin condensation. A single H1 histone protein will bind to the linker DNA here. The binding of the histone H1 to the linker DNA will help the two nucleosomes to pack to each other. So بس تحطوا هيدا الهيستون هون, guys, the linker DNA will be wrapped around the histone protein. So the first nucleosome will be near the second nucleosome. So this is the second level of chromatin condensation, where you can bring these two nucleosomes near each other, and you will convert the 10 nanometer chromatin fiber into the 30 nanometer chromatin fiber, representing the second level or the next level of chromatin condensation. Okay, so خلوا ببالكم, guys, our level is the 10 nanometer. It's the beads on a string representing simply the nucleosomes that link together by linker DNA. Once you will add the H1 to the linker DNA, you will move to the 30 nanometer chromatin fiber, and you will create the next level of chromatin condensation. Beyond the 30 nanometer chromatin fiber, the chromatin packing beyond this 30 nanometer chromatin filaments depends on non-histones. وخلصنا الهيستونز proteins, هلا بنستعمل the non-histones protein. One of the non-histone protein is known by scaffold protein. What is the shape of this scaffold protein? This is the scaffold protein, guys. This is the protein alone without the DNA. مين بتشبه هيدي البروتين, guys؟ مش بتشبه the X-shaped structure of the chromosome, duplicated chromosome with 2 sister chromatids. So نحن, guys, ليش بنرسم الكروموسوم هيك X، نرسم الكروموسوم X shape؟ ليش بنرسمهم بهي الطريقة؟ We use this X-shaped chromosome structure, guys, the duplicated chromosome with 2 sister chromatids, because the shape of the scaffold non-histone protein is similar to the X-shaped structure. Okay, so only the 30 nanometer chromatin fiber will wrap this X-shaped scaffold protein. That's why at the end, the shape of the chromosome is similar to an X-shaped structure. Okay. Since we defined the structure of chromatin, the scientists discovered that these chromatin filaments inside the nucleus, guys, they shift through the cell cycle between two levels, guys, two states. Sometimes you will find the chromatin in a dispersed state. Mainly بنشوف the most dispersed states during the S phase اللي هي the duplication, the replication of the DNA. So بنشوف ال DNA مثل كأنه مفرفط معجق كله على بعض. And in the second time, you can find the DNA, the chromatin, highly condensed when the cell shifts to the mitosis or meiosis, the cell division. This changes in the state represent the changes in the degree of DNA packaging or packing. So that's why the scientists discovered two forms of the DNA. The first one is known by euchromatin, and the second one is known by heterochromatin. So what is the difference between the euchromatin and the heterochromatin? I will start first by the euchromatin, guys. Euchromatin, it's a part of the chromosome, or it's the chromosome representing the normal cycle of condensation, decondensation, condensation, decondensation. So بتشوفوا انه شقفة من الكروموسوم او large section of the chromosome ساعة بتعمل condensation so بتلك على حالها، ساعة بتعمل decondensation so بتفك. Okay, so رح نشوف انه this segment رح يضل عندها a normal cycle of condensation followed by decondensation. The genes present in this region of the euchromatin are considered as active genes. Active means actively transcribed. So I can convert the information from gene to protein in this level. And in this region, you will find only genes without repetitive sequences. Why we need to condense and decondense the actively transcribed genes? لأنه وقت تكون مفتوحة مثل بهالطريقة guys in the decondensed structure, the DNA is more accessible by the transcription machinery and the replication machinery. So the RNA polymerase can reach easily the DNA and transcribe it into mRNA and then protein. However, when the DNA now is not used by the cell, so as أنا هلا I don't need the transcription of this gene. So what will happen to this portion? The decondensed form will be converted into condensed. So رح لكن على بعضهم لهول النيوكليوسوم. So هيك the DNA is not accessible anymore by the transcription machinery, and you will turn off the transcription of this gene. Okay? So we defined the euchromatin. Now we'll move to the heterochromatin. The heterochromatin is also a part or region of the chromosome, بس هالمرة the heterochromatin region is always in the condensed form. So this is the structure of the euchromatin, so shift between decondensed and condensed. However, the heterochromatin is always present condensed. And when we say always condensed, معناتها the DNA is not accessible easily by the transcription machinery. معناتها the RNA polymerase cannot access the DNA, so no transcription in this region. So the genes present in this region are transcriptionally inactive. Two forms, two types of the heterochromatins are present. The first one is known by constitutive heterochromatin, and the second one is known by facultative heterochromatin. The constitutive one, guys, represent heterochromatins but always present at identical positions on both homologous chromosomes. إذا بيجي بتطلع guys على الهومولوجous chromosome، إذا بتطلع على البوزيشن للتيلوميرز والبوزيشن للسنتروميرز، these region guys مش دايما عندهم exactly the same position between homologous chromosomes. So in this region رح تشوفوا heterochromatin. So telomere and centromeres are always condensed, transcriptionally inactive. So centromeres and telomeres represent heterochromatin. Because they are always present at identical positions on both homologous chromosomes, so they are considered as examples of constitutive heterochromatin. However, the facultative heterochromatin من اسمها guys بعكس constitutive, the location of these heterochromatins vary depending on the cell type, developmental stage, and from one homologous chromosome to another. دايما عم نحكي heterochromatin means transcriptionally inactive, means remain condensed. هعطيكم one example of facultative heterochromatin. One example is the bar body or بنسميها the Lyon hypothesis أو اللي بنسميها كمان X inactivation in female. شو معناتها guys the bar body or X inactivation؟ كلنا بنعرف guys إنه الميلز عندهم X and Y. However, الفيميلز عندهم XX. إذا بدنا نحكي about the doses of the X chromosome, بالmale we have only one dose. However, in female we have two doses of X chromosome. ولأنه let's say we are both humans, so we are equal guys. So مفروض لازم يكون عندنا one dose of X chromosome in male and female. So how we can equalize the doses between male and female? If you want to equalize the dose ليكونوا قد بعضهم، إذا الميل عنده 1X معناتها الفيميل لازم يكون عندها 1 active X and 1 inactive X chromosome. So how I can inactivate one of the X chromosome in female? On X chromosomes guys رح تشوفوا في one region known by X inactivation center. In this region you will find a gene known by exist gene. شو هو exist gene guys? Exist means X inactivation specific transcript. So exist gene stands for X inactivation specific transcript. It's present on both X chromosomes. But in each cell, one of this exist gene will be active. Let's say خلينا ناخد هالexample إنه this exist gene is active. If this exist gene is active, guys, it will be transcribed into RNA, non-translated RNA. This RNA will coat, بتغلف, will coat the X chromosome, turning off the genes present on this coated X chromosome. So at the end, what will happen, guys? رح يطلع عندكم one active X chromosome and one inactive X chromosome. وهيك بيصير في عنا the inactivation of one of the X chromosome and the equalization of doses between male and female. Hi, it's an example of facultative heterochromatin. ليش it's an example؟ لأنه كل مرة ممكن يصير في عنا activation للexist gene على one chromosome. So it's not always at an identical position on both homologous chromosomes. Here will vary on both homologous chromosomes. So that's why it's an example of facultative heterochromatin where the genes are transcriptionally inactive and the structure will be condensed. رح أعطيكم example guys شو إذا بدكم تقولوا consequences of this X inactivation. خلينا ناخد for example color blindness guys. In female, the color blind gene is present on the X chromosome, so it's an X-linked gene. Let's consider إنه في عنا one female carrier heterozygous with capital letter C for normal vision and small c for colorblind. When the cell divide by mitosis, تخيلوا guys انه بالفirst cell صار في عنا inactivation للsecond chromosome and in the second cell صار في عنا inactivation للfirst chromosome. So from the first cell, when this first cell will divide, all the generations will have the X with capital letter C, so normal vision. And the second cell, when divide, all the cells will have the X chromosome with a small c colorblind. So at the end, what will happen? رح تشوفوا عينها لهالمرة نصها بيكون فيها capital letter C ونص cells of the eye رح يكونوا with a small c. This female will observe normally لأنه عندها capital letter C, but it's a carrier one or heterozyte. تخيلوا guys, by chance, facultative heterochromatin turned the first chromosome off in all cells. So in all cells بأول وحدة وبالثانية طفينا أول X chromosome. So what will happen guys? All the cells of the eye will be present with X chromosome with a small c colorblind. So the female will have the red-green colorblind, no normal vision. In contrast to the first example يلي أخذنا 50-50 colorblind, normal vision. This is another presentation, guys, of euchromatin and heterochromatin. اتفقنا telomere, centromere, telomere are heterochromatin, always at identical position, more condensed, okay, and they are darkly stained. However, the euchromatin بالنص are less condensed, where you can find the gene expression, and they are stained light. عظيم. رح نخلص البرزنتيشن لليوم by defining مثل ما شفنا guys the distribution of certain sequences in the genome of prokaryotes and eukaryotes. We already studied the basic structure of DNA. Now it's time to study the distribution of the sequence in the genome. The geneticists, the scientists discovered that some sequences are present only once in the genome and others are repeated. So based on this discovery, guys, the scientists divided these sequences into 3 categories. The first one is known by unique sequence DNA. So إذا لقينا sequence of DNA موجودة بس مرة واحدة بالجينوم, so رح نسميها unique sequence. But this definition was modified. So رح نقول now إنه unique sequence DNA present in 1 to few copies. يعني عم نحكي few copies, 50 times, 10 times, 20 times. Okay, so present in 1 to few copies in a genome. بنسمي هاي sequence unique sequence DNA. However, if this sequence is present in a few to about 10 to the power of 5 copies in the genome, بنسميها sequence moderately repetitive DNA. And finally, if the sequence is repeated 10 to the power of 5 to 10 to the power of 7 copies in genome, it's known by highly repetitive DNA sequence. In prokaryotes, means in bacteria and archaea, all of the genome is present as unique sequence. ليش؟ لأنه the genome of the bacteria and prokaryote is very small, so there is no space for repetitive DNA sequence. So that's why most of the DNA sequences are present as unique, except the DNA sequences encoding the ribosomal RNA and the transfer RNA. هول الاثنين, they are present repeated several times. In contrast, in eukaryotes, بتشوفوا both unique and repetitive. We have a very large genome, so you can have both unique and repetitive DNA sequence in the eukaryotic genome. I will move more in details about the unique and repetitive. So we still have 2 slides, one for the unique and one for the repetitive. The unique sequence, guys, is sometimes also called single copy. لأنه you can find this copy once per genome, single copy in the genome. And if we are talking about diploid cell, so two sets of chromosomes, so we have two copies. But in the recent usage and the current usage of this term, the scientists slightly modified the definition. So بنقول unique sequence or single copy, it's present one to few copies in the gene. Most of the genes اللي هن بيعطونا proteins are present as unique in order simply to regulate the expression of this gene. So لنقدر نعمل عليهم control. So ما فيني أحط من الجين several copies. بحط منه one copy in order to regulate its expression. And in the human genome, you can find 55 to 60% of the sequences considered as unique. At the end of this chapter, we will define the repetitive sequence DNA, both وقت قلنا moderately and highly repetitive DNA sequence. They are repeated many times within the genome, but these repetitive sequences are arranged differently inside the genome. Sometimes, شوفوا معي على الفيجر guys, if these sequences are distributed at irregular intervals, هيك randomly distributed at irregular intervals, بنسميهم interspersed or dispersed repeated DNA sequences. In contrast, if these sequences are repeated several times in a row, so they are clustered together وعم بينعادوا وحده ورا الثانيه وحده ورا الثانيه، بنسميهم randomly repeated DNA sequences. So based on the arrangement, the repetitive sequences are divided into two classes, interspersed or randomly repeated DNA. Regarding the interspersed, guys, if the sequence is short, يعني between 100 and 400 base pair long, so إذا طولها between 100 and 400 base pair long, رح نسمي هيدي sequence signs for short interspersed nuclear element. However, if this sequence is long, between 1000 and 7000 base pair long, رح نسميها lines for long interspersed nuclear element. So guys, that's it for chapter 2. If you have any question about the composition, the structure, the arrangement, the organization of the DNA in the genome, please let me know. Thank you for listening.